The commonly used hosts were bacteria and yeast, and the vectors were called as BAC, that is bacterial artificial chromosomes, and YAC, that is yeast artificial chromosomes. So hosts were bacteria and yeast, and the vectors were BAC and YAC. Now, the salient features of human genome, their chromosome 1 has most genes, that number is 2968, and the Y has the fewest genes, that is 231, 231. Now there are some positions or locations have been identified, about 1.4 million locations where single base differences. A genetic code is made up of 3 nucleotides where single base differences, these are SNPs, SNPs. Single nucleotide polymorphism, SNPs, occur in humans. Another thing, the average gene consists of 3000 bases, but sizes vary greatly, with the largest known human gene being dystrophin at 2.4 million bases. Okay, 2.4 million bases. Repetitive sequences are stretches of DNA sequences that are repeated many times, sometimes 100 to 1000 times. They are thought to have no direct coding functions, but they shed light on chromosome structure, dynamics, and evolution. So this was repetitive sequences. Now DNA fingerprinting involves identifying differences in some specific regions in DNA sequence called as repetitive DNA. Now because in these sequences a small stretch of DNA is repeated many times. Now how this repetitive DNA being separated from bulk genomic DNA. So this repetitive DNA are separated from bulk genomic DNA that we don't want as different peaks during density gradient centrifugation. The bulk DNA that we don't want forms a major peak, and the other small peaks which we want are referred to as satellite DNA. So repetitive DNA is satellite DNA kind of thing. Then as polymorphism in DNA sequence is the basis of genetic mapping of human genome as well as of DNA fingerprinting, it is essential that we understand what DNA polymorphism means in simple terms. Polymorphism variation at genetic level arises due to mutations